Hello students, welcome to Super 7 Revision Series Part 15. Here we will discuss 3 marks questions based on DNA, genetic code and cloning vectors. Let us start with question number 1. It is based on replication of DNA and replication starts with small opening and this opening is called as replication fork. Now read first part of the question. Identify the polarity in A and B respectively in a given figure. See the diagram. See the two newly formed DNA strands. Polarity at A is 5' and polarity at B is 3'. Note important point. DNA polymerase enzyme जो DNA replication में help करता है, it always works in 5' to 3' direction. Now, second part of the question. Name the types of synthesis A and B occurring in the replication fork of DNA. The replication at leading strand, number 1 in diagram, it is continuous. So, synthesis A is continuous. While the replication at lagging strand, number 2 in diagram, it is discontinuous, means small fragments of DNA are formed. So, synthesis B is discontinuous. Third part of the question, name the enzyme that joins small fragments of DNA of lagging strand. It is DNA ligase enzyme which joins small fragments of DNA. Come to second question, it is based on messenger RNA. Part A of question, what hnRNA stands for? It is heterogeneous nuclear RNA. Note important point about hnRNA. It is a precursor of messenger RNA and it is non-functional. Part B of question. Identify A, B, C in diagram. See the diagram. Dark shaded part A is intron, light shaded part B is exon and C is poly A tail having adenylate residues which are 200 to 300 in number. Part C of the question. What are the steps hnRNA will undergo to form messenger RNA? It will undergo 3 processes to form messenger RNA. First step is splicing. Here introns are removed and exons are joined together. Second step is capping. See the diagram at 5' end of hnRNA. Mg with 3-phosphate is added. It is methyl guanosine triphosphate. It is added at 5' end of hnRNA. Third step is tailing. See the diagram at 3' end. Polyadenylated tail or poly A tail is added and finally hnRNA changes to functional messenger RNA. Question 3. It is based on transfer RNA. Part A of question. Name the scientist who called transfer RNA as adapter molecule. Francis Crick called transfer RNA as adapter molecule because transfer RNA on one hand read the code and on other hand would bind to specific amino acids. Part B. Draw a clover leaf structure of transfer RNA. Draw diagram with pencil as shown and now show 1. Tyrosine attached to its amino acid site. Amino acid is attached at 3' end of transfer RNA. So, write tyrosine at 3' end. Next, show anticodon for this amino acid in its correct site. See the site of anticodon. It is A in diagram. As codon for tyrosine is UAC which is given in the question. So, write AUG anticodon at site A of transfer RNA. Part C of the question. What does the actual structure of transfer RNA look like? It looks like an inverted L. Now come to question number 4 based on genetic code. Given below are sequence of nucleotides in a particular messenger RNA and amino acids coded to it. See the given sequence with amino acids and write 3 properties of genetic code. First property is codon is triplet, means sequence of 3 bases form a codon, like UUU, AUG, UUC, all are sequence of 3 bases and they are forming the codon. Also note, UAA is not coding any amino acid. It is a stop codon. Out of 64 codons, 3 are stop codons and these are written on side block. UAA, UAG and UGA are stop codon. Second property, genetic code is unambiguous, means one codon code for one amino acid only. You can give one example from the above given information, like GAG will code for glutamic acid and you can see it is written in the block. Now third property, genetic code is degenerate, means one amino acid is coded by more than one codon. The example of this property is phenylalanine amino acid coded by 2 codons, UUU and UUC. One more property of genetic code is given in question number 5. Explain the dual function of AUG codon. This is the first part of the question. The dual function of AUG codons are: 1. It codes for amino acid methionine. Second, it functions as initiation codon, means it initiates or start the translation process. Part 2 of the question, give the sequence of bases it is transcribed from, as transcription take place from DNA to messenger RNA. So, for given codon AUG, the base sequence on DNA will be TAC. So, write the answer. It is transcribed from TAC on DNA. Part 3 of the question, write its anticodon. As anticodons are present on transfer RNA, the anticodon for AUG on transfer RNA is UAC, and you can also draw the diagram along with the answer. Question number 6. This question is based on packaging of DNA molecule in nucleus. Part A. What is this diagram representing? This diagram is representing nucleosome. Part B of question. Name the parts A, B, C and D. A is histone octamer. It is formed by 8 molecules of histone and it is positively charged. B is DNA. It is negatively charged and it is wrapped around positively charged histone octamer and form a structure nucleosome. A typical nucleosome has 200 base pair of DNA helix. C is H1 histone. It is a linker histone which joins 2 ends of DNA in nucleosome, and D is a core of histone molecule. Question number 7. This question is based on cloning vectors, which are very important tool in recombinant DNA technology. Read the question. Draw a schematic diagram of E. coli vector PBr322. First, draw diagram with sharp pencil. Before marking the different parts, let me tell you what PBr322 stands for. P stands for plasmid. Br. These 2 letters are taken from the names of scientists who constructed PBr322. B is taken from name of Bolivar and R is taken from name of Rodriguez, and 322 is a number assigned. Now start marking as asked in the question. See the diagram and mark different parts. Marking A in diagram is ori. It is the origin of replication. Note important point about ori. From here the replication starts, and also it controls the copy number of linked DNA. Marking B is ROP. It codes for proteins involved in replication of plasmid. Marking C and D in diagram. C is ampicillin resistant gene and D is tetracycline resistant gene, and these 2 are selectable markers. Note important point about selectable marker. They are used to identify and eliminate non-transformants and permit the growth of transformants. Next, E and F marking. Marking E in diagram is BAMH1 and F is EcoR1. Both these are recognition site for restriction enzymes endonucleases. See one more cloning vector in question number 8. How is insertional inactivation of an enzyme used as selectable markers to differentiate recombinant from non-recombinant? See diagram 1. It is PUC8 or PUC8 vector. Here P stands for plasmid, UC stands for University of California, and 8 is a number assigned. Now see what is there on PUC8 in diagram. It has a LAC-Z gene. This gene is responsible for formation of beta-galactosidase enzyme. This is number 1 plasmid. It is non-recombinant type. Now let us introduce a gene of interest at location LAC-Z. See the diagram 2. Here required gene is inserted, which is shown by green color in plasmid, and now this plasmid is recombinant type and this will not produce beta-galactosidase enzyme. Now see what happens with chromogenic substrate. Note in the diagram, the plasmid 1 will give blue color with chromogenic substrate because it is producing beta-galactosidase enzyme, and the plasmid 2 will not give blue color with chromogenic substrate because it is not producing beta-galactosidase enzyme. So, by inserting a gene of interest, inactivated the LAC-Z in recombinant plasmid, and this is called as insertional inactivation and is used to differentiate recombinant from non-recombinant. Here is another cloning vector used in plants. Question number 9. State how Agrobacterium tumefaciens has been made useful cloning vector to transfer DNA to plant cell. Agrobacterium tumefaciens is a pathogen of several dicot plant. See the diagram. It has tumor inducing plasmid with tDNA, जो colored part से दिखाया गया है यहाँ पर. This bacteria can deliver tDNA into plant cell and plant cell changes to tumor cells. But how can we use it as cloning vector? It can be done by modifying tumor inducing plasmid. As a result, this is no more pathogenic, but it can deliver a gene of interest in target plant cell. Come to retrovirus as cloning vector used in animal cells. Question number 10. How can retrovirus be used efficiently in biotechnology experiments in spite of them being disease causing? See diagram number 1. It is a natural virus which causes disease, but by removing the disease causing gene, it is shown in diagram, green colored gene is removed from retrovirus and we added gene of interest. This is shown by pink color in diagram 2. As a result, what will happen? This retrovirus will not cause any disease but can deliver the gene of interest to target cell. So, that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.